This video explains how to operate CellaVision software for classifying digitalized bone marrow cells. About how to access our PC at MLTC Japan, please refer to our separate PDF manual. When you access our PC, you see 5 data files which are obtained from 5 different patients and allocated to you. So double-click one file to open. You can see cell fractions which have been pre-classified by the CellaVision. This classification is very incomplete and includes many mistakes. In this basic display mode, you can see every cell fraction by scrolling. Let's take a look. You can see every cell fraction by scrolling. Some fractions are good and some others are not good, include many mistakes. Monocytes, promyelocytes, you can see erythroblasts, and this is the end of fraction. You can change display mode by clicking these icons. We recommend to use basic mode or two-window mode in most of your work. In this two-window mode, one cell fraction is shown in each window, which can be changed by drop-down menu. Results of classification by the CellaVision are insufficient and contain many mistakes. Your work is examining every cell fraction and correcting the mistakes. Remember that the CellaVision has been designed to classify blood cells and then can't classify immature form of monocytes, type 2 blasts, and all stages of erythroblasts. Most erythroblasts are classified in the nucleated RBC fraction instead. Please work on all cell fractions including unidentified, nucleated RBC, and smudge cells. First, we work on unidentified folder in two-window mode. Since unidentified folder contains many smudge cells, we change the cell fraction in the right window to smudge cells. In the left window, choose multiple smudge cells by using a usual keyboard operation, such as pressing Ctrl key while selecting multiple cells, and drag them to the smudge fraction. Next, to locate erythroblasts observed in the unidentified folder, we change the right cell window to polychromatic erythroblasts and move all erythroblasts to the right window. Finally, we classify other cells. By left double-click, you can enlarge the image. You can see the cell list by right-click. Although the CellaVision didn't classify this cell, the CellaVision provided the second and third candidate cells. If you want to classify this cell as a monocyte, you can select the second candidate in this case or the monocyte among the cell list. Let's take a look. We continue to classify other cells. Next, we work on nucleated RBC folder. Since most cells in this fraction are polychromatic erythroblasts, we move all erythroblasts in this window to the light polychromatic erythroblast window. Then we change the light cell window to smart cells and move all smart cells in the nucleated RBC folder to the light window. And finally, we classify the rest of cells. Since polychromatic erythroblast folder contains alpha erythroblasts, you can reclassify these cells according to your own criteria. Conclusions. Examine every cell fraction including unidentified, nucleated RBC, and SMATch. Every operation is auto-saved, so you don't have to save by yourself. We will examine the consistency of key data, mainly percentage of blasts, when the bone marrow digital images and bone marrow smears from the same patients are analyzed by the same investigators. So the important point is that applying the same criteria of your own in analyzing the digital data and smears, particularly for blasts and stuff to be eliminated. This study never cared about the difference between type 1 and 2 myoblasts, stub and seg, kinds of erythroblasts, and smudge and artifacts. If you don't distinguish between type 1 and 2 myoblasts, classify all myoblasts to type 1 myoblasts. Finally, in the basic display mode, please make sure that you have worked on all cell fractions. Remember that in microscope work, you classify each cell in a second. Meanwhile, in this digital work, you can spend more time on each cell. However, we recommend that you don't think too much and classify cells smoothly as usual.